Hey guys, welcome back. Today we're going to talk about human genetics. Just as we talked about genetics in general, now we're going to focus on human genetics. I hope you're really excited about this one. So first we're going to talk about pedigrees. So a pedigree is used to study the inheritance of a single trait in a family. So let's look at the symbols. A square designates it as a male. A circle designates as a female. If it is filled in black, those are affected individuals. If there is a line between a square and a circle, that means mating. All right. And then to designate offspring, offspring are listed left to right in order from birth. Okay. So if you look here, we have the square and the circle mating, and they produce first a daughter and then a son. Okay, so the daughter is older than the son. And then 1 and 2, those are the generations. All right, so let's think of inheritance patterns of an autosomal dominant trait. So autosomal means it's on one of the genes or one of the chromosomes that are not sex chromosomes. Remember, our autosomes are our non-sex chromosomes. All right, in autosomal dominant traits, an individual needs one dominant allele, so one capital allele to have the trait. Okay. Therefore, because you only need one capital allele, you'll find the trait in every generation, and it is expressed in both males and females. And this pedigree is an example of an autosomal dominant trait. All right, autosomal recessive. All right, so for an autosomal recessive, you need two recessive alleles, or also known as two lowercase alleles. This traits tend to skip generations. All right, and the trait can be expressed in both males and females. Okay. All right, sex-linked recessive traits. Okay, so in sex-linked traits, males need one recessive allele, okay, because the trait is only carried on the X chromosome, so they only need, and they only have one X. Therefore, if their X has the recessive allele, then they're going to have the disease. Females need two recessive traits, okay? And then females can be considered carriers because they can be heterozygous. Okay, for sex-linked recessive traits, it tend to skip generations, or it's just how the inheritance pattern occurs. It is not that they're willfully skipping the generation. It's just how it occurs. And the trait is often expressed in males more often than it is in females, and it is often comes from mother to son. Well, it does come from mother to son. All right, what is a trait? A trait can be any normal characteristic. It can be abnormal characteristics. Okay, so traits include normal characteristics and disorders. All right, karyotypes. A karyotype is a picture of chromosomes, like the one shown in this image here. All right, your autosomes are your first 22 pairs of chromosomes, and your sex chromosomes are the 23rd pair. All right, karyotypes will show you your chromosomal disorders and your aneuploidy. Aneuploidy means an abnormal number of chromosomes. So let's look at some of these. To do karyotypes. So karyotypes are historically taken in two different ways. First is an amniocentesis, that is taking a really long needle and putting it through the stomach of a pregnant woman into the amniotic fluid, withdrawing a little amniotic fluid, and then sending it off to be tested. You also could, earlier on, when the fetus is smaller, you can do chorionic villi sampling. This is sticking a little catheter in to test the chorionic villi, which exists between the fetus and the edge of the uterus. And of course, in both of these, they use an ultrasound transducer to get image and make sure they're doing everything in the right place. Nowadays, they have newer ways to test, but to get karyotypes, these are the two main ways that we test for chromosomal disorders. All right, gene disorders cannot be seen on a karyotype because they occur only in faulty genes. A gene is just a section of DNA that codes for a particular protein, and remember that proteins determine traits. So a particular gene won't be found on a karyotype. All right, let's look at some gene disorders. First, we have Huntington's disease. This is autosomal dominant, so it is not on your sex chromosome. If you have two capital HH, you have the trait. If you're heterozygous, you also have the trait. You will only not have the trait if you are recessive. Huntington's disease appears after age 40. It leads to a gradual deterioration in the brain and eventually death. You can look at the two images of the brain. The WT means without, and then HD means Huntington's disease, and you can see the significant changes in the brain. Sickle cell anemia. It is autosomal codominant. This is an example of a codominant trait. Okay, sickle cell. If you have both sickle cells, you only you have the disease, and you have sickle cells. Okay. Sickle cells, SN, you're heterozygote, you have the trait, you have some sickle cells, but some normal cells. You don't often have the negative aspects of the disease, but you have the protective aspects of the disease. Because remember, sickle cell protects, having sickle cell blood cells protects you from getting malaria. It's best when you are a heterozygote. If you're a homozygote and have the disease, there's a lot of other problems that come. And then if you don't have the trait, you have normal blood cells. The problems that come with this is it can lead to poor circulation and pain, and it's predominant in African and African American populations. All right, gene disorders. Cystic fibrosis. It is an autosomal recessive disease. So you have to have two carrier parents, two heterozygote parents for the child to have it. It leads to mucus buildup in the lungs and digestive tract. It is usually fatal, though they're starting to see people in their 30s with this disease. It's rare. It is predominant in Caucasian populations. Tay-Sachs disease. This is another autosomal recessive disease. You have to have two carrier parents. It causes the breakdown of the central nervous system, leads to death by age 4. It is predominant in Jewish populations and also Amish populations. Phenylketonuria. Everyone in this class has been tested for PKU. You're tested at birth. They do a little prick of your foot. They put a little blood on a piece of paper and they send it off and check. It is autosomal recessive. You must have two genes for two of the genes to have it. What it means is you're going to have the inability to break down the amino acid phenylalanine. This results in phenylalanine building up in the brain, which can result in retardation and other problems. However, this is a highly treatable disease disorder because you can just choose to eat low phenylalanine foods. If you look at foods, they will all say if they are high or low phenylalanine or if they contain phenylalanine, and they'll say like phenylketonurics. Okay, I look on a can of Diet Coke and you'll see it. Hemophilia. This is a sex-linked recessive. Okay, hemophilia means you don't make enough of the clotting factors needed for your blood to clot correctly. It leads to excessive bruising and bleeding, especially around your joints. This is most often seen in males. Though we're starting to see it in females for the first time in the past 50 years, because it used to lead to death before that. Red-green color blindness. I'm sure we have some people with red-green color blindness. This is a sex-linked trait. It is recessive. It means you're unable to distinguish between red and green. Generally, all colors you have a little bit of difficulty, but especially reds and greens. In this image right here, you should be able to see the number 2. All right, and then on this side of this image, you should be able to see some red jelly beans and some orange jelly beans. All right, chromosomal disorders. Chromosomal disorders can be diagnosed using a karyotype, and it can involve problems with an entire chromosome, which contains thousands of genes. So, and this results in aneuploidy. Aneuploidy is an abnormal number of chromosomes, and this results from mistakes in meiosis. one probably very common that you are aware of is Down syndrome. It is trisomy 21. It's the 21st chromosome. There's 3 copies of it. You could have 3 full copies, or you could have 2.5, but you have extra of the 21st chromosome. It leads to mental impairment, okay, heart defects, flat facial features, and an enlarged tongue. Here's an image of an individual with Down syndrome. You may be able to connect that to somebody you know in your life. Turner syndrome. This is a sex chromosome aneuploidy. It means they only have an X chromosome and nothing else, just one X chromosome. So we say XO. They are female, but they are infertile, and they often have mental impairments. They also have problems with their lymphatic, cardiac, renal, ocular, auditory, skin, orthopedic growth, ovarian development and reproduction, and psychosocial problems. Okay, so there's lots of issues that occur from Turner syndrome. Klinefelter syndrome. There are 2 X's and a Y. These are male. Okay, we consider them male because they have a Y chromosome. They have mental impairments, and they are infertile. All right, gene therapies. Genetic disorders can be treated but cannot be cured. But our work through the Human Genome Project has given us a lot of information about how diseases develop and how we can make gene therapies. Gene therapy is replacing a defective gene with a normal gene. It is what we consider the future, but we will see how that goes over the years. There's lots of pros and cons to this and a lot of debate. All right, I look forward to talking to you more about human genetics in class and all the various diseases, and for you to do some projects on this information. Talk to you later. See you in class.